Welcome back to the course. In the last lecture, I tried to tell that the bakers is actually basically we call it as a food yeast, and this is produced by using Saccharomyces cerevisiae. Now, this is used for leavening of the bread, and I told you that it is a good source of nitrogen, also vitamin content. So, now I am going to discuss this process in details. The most important thing that we have the factors that affect the baker's yeast fermentation process. I told you the selection of the microorganism plays very important role. Now question comes, what should be the characteristics of the microorganism? First thing, that it should be genetically stable, which is very important, because if organisms are genetically stable, then and only then the biochemical characteristics of the organism will be same. So, and it should have high fermenting power, so that degradation of the substrate will be very fast, and then the rapid growth characteristics giving the high yield on cheaper raw materials, because the raw materials more cheaper, your cost of production depends on that. If the raw materials is cheaper, your cost of the yeast cells will be less, and then readily disperse in water, because this let me explain is very well, because this is a special characteristics we want for the yeast cells, readily disperse in water. Now, what does it mean? Suppose we have yeast cells here, and we make a cell suspension here. Now, if cells are there like this, suppose some cluster of cells are like this, and cluster of cell means lot of cells they are binding together like this. Now, so I told you this is used for the bread making purpose. Now, when you prepare the dough, suppose you are preparing, this is a container, and in this container you keep the dough. You prepare the bread, the dough you prepare and keep it. Now, if yeast are not dispersed properly, if they are not segregated, then and they form this kind of cluster, then what will happen when your dough bulge and dry it, and then when you cut the bread, if you take the surface of the bread, if you this is the slice of the bread, if you see it, then you will find there some big holes is there, some small holes are there. Because why the big holes are there? Due to presence of the cluster of cells, and due to the presence of cluster of cells, they produce more carbon dioxide and they make a big hole here. And since here you have seen the single yeast cells, it form the smaller holes. So, the porosity will be different. So, this is undesirable, and that is why one important characteristics of the yeast cells is the readily dispersion. Whenever you prepare a suspension, it should be uniformly dispersed in water. Then good appearance, because whenever we go to the market and purchase any kind of product, the appearance of the product also very important factor. So, from the appearance you have to see that whether it is good or bad. Usually yeast should look like a perfect white color, but if it is of different color, that means maybe due to some contamination problem. And resistance to autolysis, I mentioned that also, this is, you should not degrade. If you degrade it, then you will not get the rapid fermentation characteristic. It should have good keeping qualities, especially during storage. Now, another thing, very important thing that we have, that is the media, because after when you have good microorganism, now good media also very important. Now, one very important thing that we have in the mind, how you design the composition of the media. Particularly, I have taken this baker's yeast fermentation as a kind of example, that how you can, is it possible, what is the methodology through which we can design that, what are the components required for this fermentation process. Now, we already observed that molasses is the best raw materials for the yeast fermentation process. Why? The reason is that it contains a good amount of sugar, it good amount of vitamins, and a good amount of nitrogen source, minerals and other things which is required for the growth of the organism. So, we do not have any problem for the growth of the organism is not a problem at all in this, but question comes that whatever material, whatever component present in the cane molasses, whether it is suitable for yeast growth or not, that we shall have to find out. That is exactly because initially when I talk about the growth characteristics and all these things, then I try to tell you that when any microorganism grow, they require couple of things. One is nitrogen source, they require proper carbon source, they require minerals, they require vitamins, and everything has a different purpose. I told you carbon is used for the cell mass formation, carbon is used for the energy, and also carbon sometimes carbon is used for product formation if the product is other than cell mass. Nitrogen mostly contribute for the cell mass formation, and minerals and vitamins they only contribute as a cofactor in the metabolic pathway for the different enzymes. Now, here that if you look at the composition of the cane molasses, I have taken the example of cane molasses because India we have mostly the cane molasses availability because we have lot of cane production and mostly the sugar industry based on the cane. So, cane molasses is our major raw materials available in India, and if you look at the composition is like this, total solid the sugar content is 50 to 58%, nitrogen 0.08 to 0.5%, and for phosphorus as P2O5 0.009 to 0.07%, magnesium oxide 0.25 to 0.8%, calcium oxide 0.15 to 0.8%, and K2O what is that is 0.8 to 2.8 to 2.2%. But if you look at because yeast has to grow, so what we do that we take the yeast, dry yeast and try to find out what is the elemental analysis of this yeast cells, and we find the yeast cells content about 8.5% nitrogen source, phosphorus is 2.5% on dry basis, then magnesium oxide is 0.4%, and calcium oxide 0.05%, and K2O 8.25%. Now, we know that yeast cells might be containing, when it present in the fermentation media, can containing more than 90% of moisture. So, naturally the concentration will be little bit less than that in the wet conditions, and one thing apparently it looks that two things is quite less in the cane molasses. One is nitrogen content. If you consider the nitrogen content in the yeast cells and nitrogen content in the cane molasses is quite less, and phosphorus content also significantly less as compared to other things is more or less same that we have as compared to this, but sometimes we find the magnesium oxide also little bit late as compared to what is required for what is have in the yeast cells. So, it has been observed that additional material required for the molasses fermentation is like this: nitrogen 1.6 to 1.8% of nitrogen on molasses weight, phosphorus P2O5 as 0.6 to 0.8% on molasses weight, MgO 0.1 to 0.15% as molasses weight. So, this is the requirement we have calculated. Now, typical material analysis has been done, and it has been found for the production of 1 kg of dry yeast required 4.3 kg molasses, 0.9 kg ammonia, 0.3 kg ammonium dihydrogen phosphate, 1.1 kg diammonium sulfate, and 60 kg of air. So, this is the requirement, basic requirement for 1 kg of dry yeast production. Now, you can remember that initially I showed you some stoichiometry of the bioprocess. Now, I want to try to show you how from the stoichiometry we can analyze this kind of whatever that requirement we have molasses for this the Baker's yeast production, whether we can correlate with this stoichiometry. Now, you can remember that stoichiometry is like this. This is the 1 gram atom of carbohydrate. Carbohydrate, we understand that carbohydrate, the name signifies that carbon and water, that is the that should be present there. So, it is C6H12O6, that if you consider glucose and if you divide by 6, then you will get this the 1 gram atom of substrate, and then you required let us assume yn by S, that is ammonia, and yo by S oxygen. This gives the yx by x cell mass. This is the cell mass, the molecular. Let us assume this formula of Baker's is like this, and yc by S is carbon dioxide, yw by S is water. Now, if you do the degree of reductions of substrate and degree of reduction of biomass is coming 4 and 4.2, and sigma S and sigma B is like this. And we know now with thermodynamic efficiency of the aerobic process, we know it varies from 0.5 to 0.6. Now, if we assume this value in between that 0.55, then eta this will be, because we epsilon p will be 0, because we assume here, because we assume no ethanol production, because this is aerobic fermentation process. Under aerobic condition, no ethanol production take place. So, we can assume the eta equal to this is yx by S, that gamma b by gamma S. So, yx by S we can easily calculate to 0.52. Now, if you do the once you get this value, then we can find out that the other value we can easily calculate, that we can find out yc by S, yn by S, yw by S. We can all we can calculate, and we can put it in this equation like this. That and when we b value we can also calculate, then we know that 1 kg we just now we find out before that for 1 kg dry yeast we require 4.3 kg molasses. So, what we have done that 1 kg with the 443 kg cane molasses, let us assume it contains 50% weight by weight sugar. So, sugar content is 2.115 kg. Now, 2.15 kg sugar multiplied by 1000, then it will be gram. This will previously it was kg. So, previously it was kg, and it will 1000 you will get gram, divided by 342. The molecular weight of sugar is 342. 6.28 moles of sugar is there. Now, 6.28 moles sugar can produce 2 moles of glucose, and then you multiply this much of glucose. Now, this much of glucose, 1 mole of glucose contains 6 moles of CH2O. You multiply by 6, you will get this atom. Now, 1 gram atom substrate produces, now you see that our stoichiometric equation, 1 gram atom substrate produces 0.52 grams of this. If it is like this, then what we can write, 1 gram atom substrate produce this gram of cell mass. So, this gram atom of substrate produce how much? This is coming about 987 gram per gram of cell. This is approximate to 1 kg of Baker's yeast. So, what I want to point out that stoichiometrically we can also justify that how this amount of cane molasses is used for the Baker's yeast fermentation process. Now, let us talk about the stages that we have in the Baker's yeast fermentation process. One is the development of the seed culture, the proper mixing of beet molasses and cane molasses. I told you 60, 50, 50. They some cases they find 60, 40 is the best nutrient for the development of the yeast cell. In the slant culture, the yeast cell loses the fermenting power. That is, the cell undergo lysis because it passes through the different stage of growth. The time factor, that is the log phase, is the most important growth and multiplication of the yeast cells. I told before several times that whenever we do the inoculation, that we always consider the growth cycle of the cells, and this is the mid log phase and this is the late log phase, and we always do the inoculation in between these phases. So, in between that if you do the inoculation here, naturally number of dead cell will be more, so your rate of growth of the organism will be affected to a great extent. The time of growth is very important. Log phase is the most important for the growth and multiplication of the yeast cells. Laboratory slant of 24 hours age is used. Molasses must be added to the media in the regular interval of time, and seed culture is done in 12 hours. So, this is the requirement we have. Then media is taken through the manhole of the fermenter. I told you that in the fermenter, in the suppose this is the fermenter and this is the aerobic fermentation process. When we carry out this fermentation, that this is the aerobic fermentation process. So, this is air out, this is air in. So, we close this valve. If you close this valve, then what will happen? The air pressure will build up, take place. Then there will be a manhole that might be there at the bottom. You take the material out, and that is the media is taken through the manhole, and here manhole we have gate valve. I have already shown you what is gate valve, what is gold valve and other type of valve. This gate valve is used in case of manhole. Then steam is passed through for sterilizing the media, and the seed culture is added. Then temperature of the media control 24 to 30 degrees centigrade. This fermentation is done in the batch for 12 hours. In certain interval of time, oat, molasses and nitrogen, phosphorus and magnesium oxide is added in requisite form. Sterile air is added uniformly so that the media remains in contact with oxygen. The lysis of the cell occurs due to the agitation of the media. So, agitation is try to not prohibited, we try to control the agitation speed, so that lysis, because the one positive side with the positive thing with the yeast cell has very thick cell wall. So, even you increase the agitator speed, the growth will not be muchly affected, but we cannot go beyond up to certain level. Usually 200 250 rpm is good enough for the keeping the cell in suspension. Antifoam compound is necessary for antifoaming compound is necessary whenever is required. Now, material of construction of fermenter, I already explained that stainless steel containers are used. Different type of stainless steel that is available in the market. I told you the SS304, that is largely if you use for our in the homemade utensils, and in the chemical industry we usually use SS314. Also we use in the biochemical industry usually we use the SS316, because most of the fermentation process it produces lot of organic acid which is corrosive in nature. So, naturally that helps us to develop that so that the resistant property of the construction materials should develop. Now, here I have given you some instances that it should contains 12% of chromium. It prevents the surface corrosion of the tank by acid. It should contain 8% of nickel to give the austenitic structure. Austenitic structure means smoothness of the metal structure, if the surface of the metal structure is cored is a little bit rough, then what will happen? The cells will grow on the surface of the material of construction. That is undesirable. We want that all cell, in case of start tank reactor, all cells should grow on the liquid itself, not on the surface of the container. So this is requirement. 8% nickel is required to give the austenitic structure. Then 2 to 5% molybdenum, which can increase the resistance power of steel by acid, because in fermentation process acids are usually formed. So this is how we can improve the quality of the stainless steel by changing the different metal content in the alloy steel. The recovery of Baker's is separation of the yeast cell from the fermentation liquid. The fermentation broth is centrifuged, or it is washed with water to free the cells or coloring matter. The ethyl alcohol, propyl alcohol, butyl alcohol can be used to wash the yeast cells. That after separation of the yeast cells, the solution of the yeast is called the yeast cream, or it contains water. This is the process flow diagram. I can explain. This is the conical flux where the cells grows here. Then cells propagate in the inoculum vessel. Then it comes to the production fermenter. In the production fermenter, we take molasses, we take nutrients. I told you have to take a nitrogen source, we have to take phosphorus source, you have to take a manganese oxide, then air you have to take, because this is the aerobic process. After that you do the separation of the yeast cells, then you have to cool it down, cream of yeast cells, then you pasteurize the yeast cells, then heat exchanger, again you cool it, then you drum dry, grind it, and palletization and sell it in the market. So this is usually produced, this is by the active dry yeast, but I mentioned that here the centrifugation that is used for this to separate the yeast cells, but that is not in practice in the bakers in the industry, because in the lab we do it in the centrifugation, but in the bakers industry we use the plate and frame filter press, because here I can explain that this is the cloth. You can see this is the cloth that we have, fine cloth. This is fine cloth. This is the wrap on the surface, and then this is the frame that we have, and we pass the liquid like this. Wash liquid, we pass this. When we pass this, then that liquid pass through here we collect the liquid, and the solid material will trap here. The baker's yeast will trap here, and then this this is that it is plate and frame filter press. I showed you before also that frames, they punch together, and they are wrapped with the thick cotton pad, thick cotton pad, thick cotton pad, thick. So they because these frames looks like that. I showed you before also. It is just like wooden frame we have in the houses. The wooden frame is look like this. So this wooden frame, we are bunched together. We have several, and in between that we wrap the surface with this, and we can make a hole, and with the help of nut and bolt that with screw we can tight this, and this is how it is done. Then yeast cells will accumulate it here, and then we when it is totally filled up, we open this and collect the yeast cells. Then two type of yeast are prepared. I told you compressed yeast and the active dry yeast. Compressed yeast contain about 27 to 30% solid matter, and active dry yeast about 4 to 8%. I told you it is 6 to 7%, it is usually 4 to 8%, and compressed yeast can be spoiled at room temperature due to high moisture content, hence it is stored in refrigeration. And active dry yeast, it does not require refrigeration or phosphorylation, so this is used when the refrigeration technique not available. And before drying, the pasteurization is essential. Active dry yeast are packed in atmosphere nitrogen and inert atmosphere. Here let me tell you that dry yeast, the dry yeast powder, that we packed it under anaerobic conditions. Suppose you have polythene pouch here, and when you put the dry and then we pass the nitrogen here in nitrogen atmosphere, we do this. Now if we allow some air to enter into the system, then what will happen, that it contains, your cell contains, your Baker's yeast contains some kind of fats, and these fats in presence of air and moisture undergo acidity and gives a foul smell that is undesirable. Now let me show you that how the active dry yeast formation takes place. It is very interesting that compressed yeast is mixed with a plasticizer. I told you this is the binder. Binder is required, otherwise the particle cannot hold together. This is the whole problem. We use the dextrin, pectin and agar. This is largely used. Then it is passed through the extruder. Extruder, you can see that this is the screw conveyor. This the yeast is coming and screw conveyor is coming, and it passes through the small orifice. Can you see it? This is called extruder. That is going like this. And this goes in the form of thread actually, and then yeast cake block will be obtained. You can form a block, or then you can cut it, because this thread you can cut it here, so that you can have small portion. Then you dry it. When dry it, then it forms a preble. The small preble formations will be there. That is why we have seen at the initially the active dry is that is not in the exact powder form, but in the pebble forms. And then it is sent to the cold storage of room, carefully dried, and the contamination contaminants during the Baker's yeast production. Acidic pH is maintained. Media contains lactic acid. Lactic acid is used. Lactic acid used as a preservative. Acetic acid bacteria and some putrefying bacteria produced are major contaminants in this particular process. Here I want to mention the putrefying bacteria. In the day-to-day life, we know that when you go out and some dead animals is throwing in somewhere place, we get very foul smell, and this foul smell is mainly coming from due to the presence of the putrefying bacteria, that they produce a lot of protease enzyme. They degrade the protein molecule, and when they degrade the protein molecule, they produce a different type of organic acid, and different type of organic acid, they are responsible for the formation of this foul smell. So this is exactly what is mentioned, that that putrefying bacteria produce are the major contaminants and detected by odor. So you can have the foul smell after that. And while Saccharomyces cerevisiae strain has less fermenting power, hence contamination should be avoided. This I showed you before also, that this you can find out in the microscope very easily. Suppose your industrial microorganism is looking morphology is like this. So if when you observe under the microscope, if you find that other yeast cells are like this, so you can visually always differentiate that this yeast is different from this yeast. So you can easily find out the yeast contamination in if this is considered as a this we consider as a industrial yeast, and this is called wild yeast. So wild yeast is very slow growing. So naturally that will affect affects the quality of yeast through a great extent, and fermentation process to a great extent. All pipes as well as the tanks should be properly sterilized. Air pipe contains cotton wool, and air is passed through the pipe to sterilize it. Now, quality control test also very important as per the baker's yeast fermentation is concerned. The solid content, because here I want to point out very important thing, that any biochemical industry, that quality control test, 3 different sections is very important. One is the raw materials, another process, another is the product. The raw materials, if the raw materials is not properly maintained, then your product will not be proper. So, raw materials, you have a chart that you find out what exactly raw materials you want. So, when you use the raw materials for the production fermenter, you have to check thoroughly whether it is coming within this range or not. And then you have to go to the process. In the process, you have to find out whether you are maintaining proper temperature, proper pH, proper operating condition, dissolved oxygen concentration, and other the antifoam oil, so that you get the proper product. And also quality control test in the product, you have to have different composition, whether it is whenever you, because as per the Food Product Organization, it is mandatory that you have to give the composition of the food whenever you do any marketing. So, naturally that until unless your raw materials is quality is maintained and process control is not maintained, you cannot expect the better quality of product. So, this is done on the basis of solid content, color, appearance, dispersion is water, already I mentioned, nitrogen and phosphorus content. Fermenting power means that how quickly it produces, it ferments the substrate. That is very important. If the high fermenting power, your time required for the fermentation process will be less. Otherwise, it will take longer period of time. Packaging weight and baking test. Baking test means how much carbon dioxide is produced. That that also that is very important. If it produces carbon dioxide very slowly, then incubation time for the bread making industry after the dough preparation, that will be very high. That is undesirable. So, so food yeast, this is also considered the food yeast. This is the food yeast means this is called neutralized yeast. It is propagated essentially for food purpose. Because here I want to mention that I think a couple of years before, the yeast is considered as a good source of vitamin B, because doctor they recommended this yeast tablet for when we have some deficiency in our body, that the vitamin B is recommended. Now, it is usually for refined and handled under superior condition. Food yeast has the high food value. It should have the following property: high protein content, vitamin for B complex, and it should have acceptable color and agreeable flavor, high growth characteristics, and cheaper raw materials, easy recoverable. And here I want to show you one small problem. I hope this is very interesting as per baker's industry is concerned. The problem is very simple. One baker's yeast industry produce 1 metric ton of compressed yeast, Saccharomyces cerevisiae per day, using cane molasses as a raw material in a chemostat. Chemostat means continuous start tank reactor. The compressed yeast content 70% weight by weight moisture. Mu max, Ks and Yx by S values of the yeast, this is wrong, yeast are 0.5 hour inverse, 2 gram per liter and 0.5 gram per liter respectively. Cane molasses contains 45% weight by weight sucrose. Initial substrate concentration of the fermentation brought 200 grams per liter. Compute the following: minimum doubling time of the cell, total total amount of cane molasses required, which is largely required in the fermentation industry, and volume of the fermenter and maximum cell mass productivity. 1 metric ton of compressed yeast means 1000 kg of compressed yeast per day, and since it contains 70% moisture, now we can assume it contains 30% total solid. So, if you multiply it by 0.3, you will get 300 kg of dry yeast per day. We know that minimum doubling time is that ln2 by mu max. So, you can easily calculate this, and the amount of sugar required you can easily also calculate if you know this the 300 kg of that cell mass you have to yeast you have to produce, y x by s you know, that is a gram of yeast cell produced per gram of sugar, then this is 0.5. So, you required 600 kg of sugar. If I assume 95% conversion efficiency, actual amount of sugar required is 631.5 kg, and if since molasses contain 45% of sugar, if you divide by 0.45, you will get 1.4 metric ton of cane molasses required. Now, if you want to calculate the volume of the fermenter required, we know tau CSTR equal to S0 minus S by minus RS equal to, I can write like this, and we calculate the mu max value. From the mu max value we can find out the x value, d max value we can find out. From d max we find out the s value. S value means under what is d max? The d max as the dilution rate when we will get the maximum cell mass formation, then maximum rate of cell mass growth, and then at that condition what is the substrate concentration we find out, and this is the cell mass concentration, and from that we calculate we find out minus RS value, what is the rate of substrate consumption. This is coming about 81.9 gram per liter per hour, and from that you find out tau CSTS space time for this is 2.22 hours. Then volumetric feed flow rate is substrate required by initial substrate concentration. This is coming about 0.13134 cubic meter per hour. So, volume of the fermenter is coming about 0.2329 cubic meter. Maximum productivity we can easily calculate. We know D max, we know the x value. If you multiply with this, we can find out what is the maximum rate of cell mass production. So, this is all about the Baker's yeast fermentation process. We try to discuss the process in details and try to also give some mathematical analysis of this particular fermentation process. Thank you.